Hello, my name is Atharva Valanju, and today I'll try to explain the phenomenon of bioluminescence and give you all a simple experiment that you can all perform at home. So, bioluminescence is the process of production and emission of light by living organisms. Now, most of you might have seen bioluminescence in a firefly, but did you also know that bioluminescence is exhibited by a variety of microorganisms, which all have their unique way of emitting light? For example, we may come across a tiny microorganism known as vibrio fisheri, which is found in the Hawaiian bobtail squid. Now, wouldn't it be interesting if I told you that you could grow your own bioluminescent bacteria sitting right at home? Well, all it takes is a few steps and some cheap equipment which you might easily find. First of all, you will need a squid. Don't worry, it's not a live one. You will need one which you can find at your local fish market. But you have to make sure that it's not frozen, and preferably it shouldn't be washed, though that's not a necessary condition. Next, you will need a few cheap apparatus, like you would need agar medium and you would need Petri plates. I would say that in our current condition, you would find these things on Amazon. Now, let's go on to the experiment. First, you will need to incubate your squid, which you can easily do by placing it in a saltwater medium overnight. Just take the squid, put it in a bucket, fill it with salt water, leave it overnight. Okay, so after a day or two, you will see that the squid will have developed a few bright spots on it. Now, what you have to do is you have to take a toothpick. You can try to heat sterilize it on a flame and then make sure that you touch the toothpick lightly on those spots that you see on the squid, and then try to transfer that medium onto the Petri plate that you have taken. You try to transfer the medium, try to move it in a streaking pattern so that you will be able to see those bioluminescent bacteria in a much better way. And voila, you're done. You just have to cover those Petri plates with a cover and keep them in a safe, secure location for the next 2 and 3 days. On the third day, you would easily be able to see a bright glowing patch of bioluminescent bacteria in your Petri plates. And that's all there is to it. So some of you might be wondering that if we initially had a bacterial colony in our inoculum, shouldn't it have glowed from day one? Well, there is an interesting phenomenon which takes place here, which is known as quorum sensing. Let me explain this with the help of an analogy. What if I told you that I wanted to light a football field and you gave me one light bulb? Well, wouldn't it be useless? You would require at least hundreds of light bulbs to produce a light good enough to light a football field. A similar mechanism takes place in our bacteria. Only when a certain population density threshold is met does the bacteria produce light which is visible to the observer. The squid is a host of the bacteria because there exists a symbiotic relation between the two, where both of them mutually benefit each other. The enzyme which helps in the process of bioluminescence is luciferase, which is the same enzyme which helps in a lot of bioluminescent reactions, like the one in the firefly. Now, let us learn about the applications of bioluminescence. Did you know that in olden days, miners used to catch fireflies in their lamps to light up the way? Bioluminescent organisms also have an application in the medical field. In the American Civil War, certain soldiers used to have wounds which would glow. At that time, it was described as angel's glow. But later on, in scientific studies, it was found that these wounds had a certain soil bacterium on their outer surface, which produced antimicrobial substances which helped in a much faster recovery than usual. Nowadays, bioluminescence is used to detect water toxicity. When certain harmful chemicals come in contact with the bacterial colony, their light output decreases considerably, and therefore we can trace the effect of that chemical. It is not a distant future indeed to think that street lamps and other light sources may soon be replaced by plants with glowing leaves. With the powerful presence of genetic engineering and DNA recombination, it might soon be able to engineer plants which are able to produce the enzyme luciferase and thus glow. Imagine the amount of energy that would be saved and would be transformed into green energy. Thank you.